I'm Dr. Earl Beecher, your host. Today our guest is Christopher Hills. Dr. Hills is the president and chairman of the board of Microalgae International and of the Light Force Spirulina Company. And we are at the Light Force Spirulina Research and Development Center near Palm Springs, California. Dr. Hills, spirulina has become of great interest to the public recently. I wonder if you could tell us exactly what it is.
Well, spirulina is a microscopic plant that floats in the lake or the river or the sea.
So this is a plant that floats on the top of the water and gets the direct sunlight?
Some species of spirulina float on the top and some sink to the bottom. There's about 25 different species of spirulina. Some have flotation and some sink. So some have to be disturbed by currents and brought to the surface to get light. We don't use that kind of algae, although we do culture them here. We have every strain in the world here. But we tend to culture the kind that float to the top, because it's easy for us to agitate them with paddle wheels and air bubbles and things like that, and that means that they get the light when they float to the top.
Now, I understand there's something like 10,000 species of plankton. Spirulina was one particular strain?
Yes, spirulina is one of 10,000 species of algae.
And there's at least 25 of those?
Yes.
Now, why have you concentrated in the algae?
Well, at first we began to study Corella algae, which grows so rapidly. You know, that's the kind that gets in your swimming pool, and you can't stop it. It's very virile. So we thought, that is Dr. Nakamura and I, he was instrumental in teaching me about the potential of algae when I was looking for a world food that could feed the hungry. Something that could grow rapidly and in great tonnage and anywhere on the planet. And so Corella was the biggest candidate at that time. But Corella is very tiny, and it can go through any filter that you can devise. So it is very difficult to harvest without expensive machinery. And how can you feed the hungry if you can't take centrifuges and so forth to where the hungry people are? So we decided to look for another algae that could be easily harvested with cloth or with simple filters, sieves. So we found spirulina.
Now, what is so great about a tiny vegetable like that?
Well, we found that spirulina was even much better than Corella, because Corella has a cellulose wall that when it's difficult to digest cellulose in your stomach. It goes through just like grasses and so forth. Any cellulose doesn't get broken down by your digestive juices. But spirulina doesn't have any cellulose in it. It's got what they call a mucopolysaccharide wall, which is a kind of a fibrous wall that's made up of sugars, different kinds of sugars. And so we found that it was 95% digestible, which is almost unheard of in a food. And then gradually we found out that spirulina had, 71% protein, higher than any other plant in the world. Then we found out that spirulina had beta-carotene, which is now being considered as a wonderful way of avoiding getting cancer, because this has been published now in several medical journals, that those people who get beta-carotene out of eating the cruciferous vegetables like spinach and lettuce and all those things somehow avoid getting cancer, or the cancer ratio is lessened in those people. So then we found it had all kinds of pigments in it, not only chlorophyll, but phycocyanin and zeaxanthin and all kinds of pigments which your liver ordinarily makes the same kind of pigments, just like your blood has a pigment called heme in it, hemoglobin.
Yes.
It's a very similar pigment to that.
So the spirulina is vegetable protein.
It's a vegetable that is almost like an animal, because it also makes one thing that no other plant makes, and that is a starch called glycogen, which is in your muscles and is manufactured by your liver, and that's what gives you, the athlete, the energy to run. And no other plant makes glycogen except the blue-green algae. So that's a remarkable thing, that it has 0.5% of glycogen in it, which is a very large molecule. Mostly the plants store their energy in ordinary starches, like cornstarch and so forth. But spirulina stores its energy in the same kind of starch that humans and animals use.
When you say vegetable protein, that's primarily amino acids?
Yes, it has over 18 amino acids, possibly nearly 20. It was thought at first that it was low in methionine, but it was found afterwards that it was the highest in methionine of any food.
Is that a special amino acid, methionine?
Methionine is the one amino acid which you have to look in a protein to see if it's short, because that's one of the very essential ones. There are 8 essential amino acids, and spirulina has all the 8 essential ones. And there was a mistake made at the beginning of the research on spirulina, where the sample that was actually grown was low in methionine, so throughout all the scientific journals they went parrot fashion that it was low in methionine, and nobody knew anything really, but actually later on it was found that it was the highest in methionine.
Now, is that unusual?
Yes, it is, because these are what they call the sulfur amino acids, which help to metabolize energy. And cysteine is another one. It's good for your hair. In fact, one of the sources of cysteine is hair, and that's where it comes from. In China, they cut off all their hair and they make it into cysteine amino acids. That's why it's scarce.
And people eat it?
And people eat it. They don't know.
I've never heard that before.
Well, you're not exactly eating hair. You're eating something that comes from hair.
It's produced from it.
Produced from hair.
I see. What else besides amino acids is in spirulina?
Oh, my gosh, there's such a tremendous number of things in spirulina. When you look at the specification, it just blows your mind. It's like an unbelievable thing, and that's why some of the people can't believe it is real. Some of the people
because
who are
it's natural and it's there, is that it?
Nutritionists are not used to this sort of kind of nutrition.
I see. Is it because the nutrients are placed in the water?
Yes, you see the nutrients like selenium and cobalamin, B12, it's the highest source of B12. It's the highest source of beta-carotene, which is, I don't know if you know, but there's been several accounts now in medical journals, and the famous Westinghouse study, or Western Electric study, which has determined over 20 years that people who got more beta-carotene through eating green vegetables somehow affected the immune system and did not get cancer, or didn't get as much cancer as those who didn't. So now beta-carotene is a very important thing in our life, and beta-carotene has to come from natural sources in order to have this effect, because synthetic beta-carotene doesn't have the same effect.
Now, if I wanted to eat spirulina, what form is it in?
Well, you can have it in tablet form, which is just made in the powder made into tablets, or you can eat it in powder, which means you can mix it in drinks or orange juice or smoothies or soups. It makes excellent soup. Or in bread you can mix, or biscuits, or cookies, or sweets, or jelly beans. Or
when you start mentioning cookies, is it made into flour?
Yeah,
it's a powdered form.
Flour. All we do is take the water out of it. It's a fresh vegetable. It is still a raw vegetable. It's never been cooked, never been heated. We don't dry it at any great heat. We take the desert air and we put it into a spray dryer. It's under a vacuum, so at very low heat it will dry. It doesn't go above 90 or 100. We can dry it at that temperature. And it takes the water out of it, and you're getting a raw vegetable, basically. And all you do when you add water to it as you reconstitute a raw vegetable.
Now, the children of Israel lived on this for 40 years in the desert and called it manna and made it into bread. And I think you're going to show us some of this a little later so that we can see how they might have done it. And also the drying plant. We're going to visit that and see some action there. You've written a whole large number of books, Dr. Hills, that I think are of interest. I see books on light. You seem to be quite concerned with light.
Well, that's how I got into it in the beginning. You see, I did many years of research into light and its effect on human beings and on matter itself. And when I met Dr. Nakamura, I was interested in harnessing the sun's light. And so when he was a microbiologist that had been appointed by the Emperor of Japan to discover a way of feeding the Japanese people during the war, because they were completely shut off with American submarines all around, and they couldn't get supplies, and so they had to find a way of feeding themselves. And so he was then researching chlorella when I met him, and he was one of the leading experts, and he'd been with NASA and all sorts of, and to Russia and everywhere with chlorella, and so we founded
Well, this was a considerable time after the war.
Oh yes, about 1960, and then we formed the Chlorella International Union, it was called, and then for several years we got about 300 scientists around the world who were interested in chlorella. And then, but we found that chlorella was such a small organism, it couldn't be harvested efficiently by hungry people, because they don't have the expensive machinery that's needed for that. So we found spirulina, and that was such a wonderful discovery that we could harvest it anywhere on the planet without expensive machines.
How did you stumble onto spirulina? Were you just going through different strains of
Dr. Nakamura was staying with me in my house in London for 3 months, and we were working on various production techniques for chlorella and growing it, and we built model plants out of plastic and all that sort of thing. And then he happened to read an article in a London newspaper, and he saw that this spirulina could be harvested with nets or mesh. So putting cloths in the water, the natives were taking it out and then dumping it in the sand and drying it. And then they'd scrape off the sand at the bottom when it was dry like a cake. And so he rushed downstairs to me and he says, look, this stuff, this is an algae, and it's 70% protein. Higher than chlorella.
So it was an article in the London newspaper that alerted you to spirulina?
So we got so excited because this was the answer we'd been looking for years and years and years. Something that would grow as fast as algae grows, something that could be harvested every 3 days, something that could be harvested by simple people once they knew how to grow it.
How fast does algae grow?
Well, spirulina grows very fast. Chlorella grows a little faster, but not that much. In 3 days, it can replace itself. Now that's
100%?
Yeah, if we have a pond and we take a third of that pond out, in 3 days that third will grow back again. So if the pond has, say, 10,000 tons of medium, it has 4 tons of algae in it. It's when we take the dry weight. And so if we take a third of that, it means we can get a third of 4 tons every 3 days. See, and that's very rapid, because if you grow soybeans, which is the heaviest crop, you can get maybe 4 tons to the acre in 1 year. I'm talking of 12, 20 tons, maybe 25 tons. In fact, very efficiently, if we wanted to grow spirulina like pharmaceutically, that is in a perfect situation, not in a lake or in nature, we could grow 600 tons a year on the 35 acres that we have here.
That's what I was going to ask you, how large a facility you have here and what maybe some of your plans are for it.
Well, we have 300 acres, so we've only got 35 acres of ponds at the moment. That's all we would need to grow 600 tons of algae a year, but if we expanded, we could grow 300 acres of ponds, and then if we could get the property next door, which is 500 acres, and we could grow 1,000 acres, and then you'd be able to see across the desert a whole green
desert
lake,
blossoming.
as it were. The desert
How many people would that feed?
Oh, hundreds of thousands. It's a very concentrated food. In fact, I could feed the world already. Just give me one African lake. There's enough spirulina in one African lake that's 12 miles long by 7 miles wide to feed all the hungry that we have.
Well, I keep seeing these tapes on television, these programs about the starving people in Africa.
They're right just a few miles from the same lake I'm talking about. But you see, I've been in Africa to Kenya, and I've had an office there for a year, and I've had scientists there in my lab, but they won't let me farm the lake because they say it might take away the food of the flamingos, which eat the spirulina. And they need the flamingos to attract the tourists. So they'd rather have tourists than have people fed.
And so we see these appeals all the time to feed the people, and yet we have the
answer.
The answer, and they ignore it. Speaking of feeding and some of the worldwide organizations, you have 2 copies of your Enlightener. One is from Japan, I was noticing. This is the magazine that the Light Force company puts out, and I can't read it, unfortunately. I don't read Japanese that fluently.
That's the May issue of the Enlightener.
And then you have one here that I think is very interesting. It has Dennis Weaver on the cover, and this is the new issue of the Enlightener. Would you share with us what you're doing with Dennis Weaver?
Well, Dennis flew up to see me about a month ago because he got excited about spirulina when he heard that we were trying to feed the world with it, because he's into a similar trip with his organization called LIFE, which is Love is Feeding Everyone. And he gets donations from Safeway and all kinds of stores that have rejected food that the time has expired on it, but it's still good food. And he collects it in trucks, and his organization feeds about 800 people a day on wasted food.
In the Los Angeles area?
In the Los Angeles area. So he's doing it at the local level, and we're doing it at the global level. So he got so excited that he thought spirulina could really work its way in through his local level. So then we got talking, and I've been to see him in his home, and we talked, and I've invented some special soups for hungry people, like using up all the milk powder that's sitting in caves and costing the American taxpayers millions of dollars every day just for the storage. I'm trying to get that to be donated so we can spray dry it with tomato powder and rice flour to make a special nutrition soup for the malnourished in the hungry places in the world, and also in the United States. Make a soup kitchen type operation.
Putting spirulina in with these other ingredients.
Putting spirulina in there. Yeah, we can't put much spirulina because it's very concentrated, but you don't need much spirulina in it.
No.
But you mix it with tomato and milk powder and rice flour and spirulina, you've got one of the most fantastic tasting gourmet soups. And it's cheap, because tomato is cheap, rice flour is cheap. I can get 12,000 tons of rice flour now, are very cheap. And if we can get the rice flour, if we could get the spirulina along with the milk powder from the government, we'd be able to feed hundreds and hundreds of people very cheaply.
I understand when you said that you were feeding people all over the world, as well as Los Angeles, that the Light Force company had sent over quite a major donation of spirulina to help the people in
we've donated
Lebanon.
about $2.7 million worth of spirulina to hungry people around the world, or to facilities that create food for hungry people. And we've shipped it to Lebanon during the war there, and Mother Teresa and El Salvador. We usually deal through either a church or a mission or the Emergency Relief Fund and that sort of organization, so that they will take it and distribute it to all the people and give us documentation that it has been effectively distributed.
I understand why you got into spirulina, because you wanted the Malthusian theory, which said the world can't support the population. We found an answer to it.
It's nonsense, because
the answer is...
It is true, that theory, if you assume that most of the world is desert anyway, and you can't grow anything in deserts, or that there's a water problem. You see, well, spirulina will grow in wastewater. it'll grow in polluted waters, and it'll clean the water. It cleans the water. Of course, it cleans the water. Now, all these lakes that are full of algae which are polluting the water, if they harvested the algae, the water would be beautifully clean. But what they do is let the algae die, and so it sinks to the bottom and gets rotten and pollutes the lake. But if they harvested it and put it on their fields, they'd have incredible fertilizer. It's a way of cleaning water, not polluting it. So now the beauty of spirulina is that it grows in almost any kind of water. Seawater it'll grow in alkaline waters that are in the deserts and that you can't use for agriculture, and that sort of thing.
Dr. Hills, this book that you have here, The Secrets of Spirulina, I understand it contains a lot of the Japanese research that's been going on.
Well, I edited the book, and because it is medical research, I'm not allowed to sell it because I sell spirulina. So the FDA have said that is part of my labeling if I sell that book. So you have to buy that book from another publisher.
I see.
And it's a very important book. I think it says it's written by Japanese professors of health and nutrition at various universities, and it's very interesting what they say.
They tell the health benefits that people can receive through eating this particular plant?
Yes, there are 15 doctors there who, of course, have 15 different things they've used it for. Everything from hair growth to you name it. We can't make that kind of claim. And of course, they even make the claim that beta-carotene is good for cancer, but I can't make that claim because I'm selling spirulina.
I see. There's another element of spirulina that I wanted to explore with you. You have a relatively new book here on the joy of slimming.
Mm-hmm.
Do people use spirulina to lose weight?
They certainly do. They find it gives them a lot of energy when they're fasting or not eating. They feel alive in their mind, they feel full of energy, and they don't feel they need to eat food to get energy. Another thing it does is it tells the brain, when you put it in your stomach, that your hunger pangs have been answered.
So when you feel satisfied, you don't have to eat a lot of other additional food.
If you take it when you're hungry, it somehow shuts off your hunger signals. And of course, there's all kinds of theories about how, but it's a phenylalanine that tells the brain that. But I mean, I don't necessarily buy all those theories.
Could this be because you've got the proteins and you've got the vitamins and the trace minerals and everything chelated in natural proportion in this food so that you don't need a lot of other things?
I think that's the reason, because in the natural environment in which the algae floats, it is absorbing all the selenium and all the rare minerals and things that you need. It's high in potassium, which of course is needed for high metabolism. There's so many things in spirulina that actually go directly to the cells in the blood. That is, the blood is the carrier of all the nutrients to the cells, and there's certain sugars like rhamnose, which is a very rare sugar. It's almost nearly 10% in spirulina. And if you wanted to buy rhamnose, you'd have to pay $730 a kilo for it.
Oh boy.
So, and spirulina's 10% of that, that's $73. The spirulina is worth just for the rhamnose in it, and forgetting all the rest. But people object to paying even $30 for it.
Now this is the spirulina growing, and you see it's pretty thick. And when my hand goes 1 inch under the water, and you can't see it, that means it's ready for harvesting. This is even quite dense, as you see, and this optical density can be read by the computer in the laboratory, and we can control exactly when to harvest from what the reading is on the computer inside the lab. And also reads the temperature. At every 10 minutes it reads the temperature and the pH. And this particular medium has got baking soda in it, which is a natural condition under which it grows in the Great Lakes in Africa. And you see, this is done by the bubble system, which means it's agitated and kept in motion so that it keeps coming up to the sunlight for a shot of sunlight, because without that sunlight it doesn't grow. So this bubble system is what we use for inoculation seed cultures, which we inoculate the main ponds with. The main ponds use a paddle system for moving the cultures around because they find that that's better in a bigger pond. We can use the paddles. Now, this algae is grown up from a very small test tube to a smaller pond, and then a bigger pond, and then a bigger pond, and then this size pond, and then it goes to another pond over there, which is 600 square meter pond. This is a 200 square meter pond. And it comes from a 25 meter and a 50 meter pond. It's the fastest growing plant on the world. See, it replaces itself in 3 days. Can replace itself in 3 days. So if we had 1,000 acres, just imagine seeing over the desert there, as far as your eye can see, green ponds like this. And we harvest a third of it every 3 days. And then you see you're getting into numbers there that can feed the world. But you got to have, the bigger you get, the more faster it grows, because the more seed culture you have, see? So this, when we put this into a big pond, and we're going to see in a minute over there, over an acre in size, then of course when you take a third of that, you've got the other 2/3 are growing that much faster because there's more of them.
How deep is the pond?
This is 8 inches deep, 8 to 10 inches deep, but it evaporates very quickly in this heat. This sunlight uses up about a centimeter per day of water just from the sun's light. So we have to replace that every day. Otherwise it will be dried out in a few days.
How often do you have to drain it completely and start over?
We never drain it unless we're going to clean the pond.
Yeah.
And then we get a little sand blowing in and some sludge. If we harvest it, we'll transfer this culture to a bigger pond, and then they'll empty it, and then we'll clean the pond.
I see.
And we keep reusing the culture medium over and over again. Now, this is beta-carotene algae growing in here, and as you can see, and I put my hand in, it's about ready for harvesting. And I get an inch under the water, you can't see my hand. Now, before we harvest it, though, we will deprive it of nitrogen, and it will then start to grow beta-carotene to protect itself against the sunlight, as it is over there. See, it's been a few days without nitrogen in the water. That means that it gets that yellow and even more, even a red color, and then we harvest it and we take the beta-carotene out with the algae, and you can eat the algae and the beta-carotene together because it's perfectly edible. We're also doing research with this particular algae for oil bearing, since it also produces 29% lipids, oils. And the Department of Energy has been very interested in using it as an alternative as against the OPEC blackmail. So if we could grow a few thousand acres of this algae, we might be able to produce enough oil that we can use as a substitute for the fuels that we're importing. But that's a long way into the future. It'll be about 2 years before we get a standardized product that is going through our spray dryers that we can give you to eat. We could give you a few pounds of it now to eat, but to produce it on a big scale takes a few years planning. Special kind of harvesting machinery has to be designed, and it's growing in 30% salt water, you see, which is, this algae comes from the Dead Sea, so we have to take the salt out before we can give it to you to eat. And once we take the salt out, then the pressure in the cell will burst because it's built, the osmotic pressure inside the cell is built to withstand that salt. So you can't just take the salt out or you lose the contents of the cell in the water. So we have to take it out by centrifuge and catch it in a way where we keep the beta-carotene and the lipids in the cell. It's quite technical. It hasn't been done by anybody yet, effectively. We're working on it, and our Japanese partners are working on it, and our Israeli company is the most advanced in the world. They have patents on it. We have a $2.73 million project on this in the Negev Desert in the Red Sea. And it's been done in conjunction with the Weizmann Institute. And we're doing it here, though, without the benefit of that institute. We're doing our own research, American research. Dr. Beecher, I'd like you to meet Sebastian Thomas. He is from South India, and he's been working on spirulina growing before he came here, about 5 years, working and trying to grow it in agricultural villages to solve the world hunger problem. And he came here, and we brought him on board in Kenya. We sent him to Kenya, and he's staffed our laboratory there for 6 months and came back with a culture that I discovered in Lake Bogoria, and he pure cultured it. And I'd like him to tell you what a pure culture really is, because it takes a lot of love to make a pure culture.
Sebastian Thomas.
That's right.
How do you make a culture?
What we normally do is we go to different parts of the world where spirulina occurs naturally. And most of the spirulina is seen in soda lakes where there is a lot of bicarbonate. And there are some natural lakes in Africa, and most of them in Kenya, like Lake Bogoria, Lake Elmendata, Lake Magadi. So we go to these lakes and collect some sample, the lake water, and bring it to the laboratory. There we put them in special medium which encourages only spirulina. Because what happens when you collect a sample from the lake, there will be different types of algae. There will be other types of algae, contaminants, which we are not interested in. So we get a pure culture.
You use a certain medium, you're talking about certain kinds of nutrients?
That's right. We have different types of chemicals, sodium bicarbonate, sodium nitrate, almost all the elements which a normal plant requires, like any other land plants. So these algae also require different types of nutrients. And we make a medium which will promote the growth of spirulina, and at the same time, which will not allow the growth of other algae at the same space.
Just like you go to the hardware store, you buy fish emulsion and all that stuff to make your plants bloom.
Yes.
The algae are the same like ordinary plants.
It is just a plant.
It's a plant,
It's a plant,
we can
yes.
stimulate it by giving it what it likes best to grow. And
I see.
He's a specialist in that area.
Dr. Hills, are each one of these a different strain of spirulina?
Yes, they're all from different lakes in different parts of the world. Now, Sebastian is an expert at culturing this. You see, he makes a pure culture, which takes about 6 months. And this represents a pure culture of all these different algae. You see, if you focus on that, you'll see it's growing on an agar from seaweed.
What's an agar?
Well, an agar is
It's a solidifying medium. It's the same medium, the water medium, and when you are agar, it solidifies after a certain temperature.
Because that doesn't look fluid.
No, it's solid.
It's solid. So what it gets, if you keep it like this, it gets a lot of surface area. And when you streak the algae get the whole surface area, and the growth is very slow. So in these tubes, you can maintain one culture for many months without subculturing.
How do you know that's all just one strain and not a mixture?
When you get a culture from the lake, it will have different types of algae. So what we do is, when we get one culture, this is special media, as I told you before, which promotes spirulina growth. We put the sample inside this flask, allow it to grow for a couple of days so that spirulina, along with all the other algae, will grow in this. Then what happens is, we take one drop from this under aseptic conditions in this chamber. We streak it on a plate like this. It's the same agar as you saw here, but this is a petri plate which gives a lot of surface area. And you take one drop of the culture and streak it all along the surface. And what happens is, each algae has got peculiar colony characters. They will spread out, and spirulina may form colonies over this side, and you'll get colonies of chlorella or different types of algae. So they will be set apart from each other.
Pure strain, we've spent like last year $2.2 million getting a pure strain of a particular culture in Bogoria, Lake Bogoria in Africa, which he was there for 6 months doing that.
Was this a strain of spirulina?
Yeah,
Spirulina.
yeah.
But it had many other types of algae also along with it when we isolated it from the lake.
Yes.
Then we had to purify it. And this one is very good in this climate because we have tested this algae. This is one of the algae which can grow at low temperature and high temperature.
And we've specially adapted it. You see, we challenge it. We put it in the refrigerator and kill it off as much as we can. And then there's one little remaining one. We snag that one out, and we breed from that again. And then we take and we kill it off in the cold again. And then if there's one remaining one, we save him and we grow him out again.
This sounds like it'd be very time-consuming.
Yes, it takes
many
months.
months to get a pure culture.
Yeah. And then we eventually finish up with one who will live in the Arctic Circle. You see, normally it's a tropical algae. It dies at 50 degrees Fahrenheit. We have to grow it here some nights at 40 degrees Fahrenheit because we're out in the desert in Palm Springs. And this is the warmest part of the United States, but it still goes down to 40 degrees. And in most of other parts of the United States, it freezes, like in Florida, where you lose a citrus crop. We couldn't grow algae there because we'd lose our crop overnight.
I see. So after you've got it at this point, then what do you do?
We look it under the microscope and see which is Spirulina. And again, under this hood, under the aseptic conditions, we pick out that colony, which is Spirulina, and stick it again on a plate like this to see if there is no more bacteria. And finally, what we do is the same culture, we streak it on medium which can promote the growth of bacteria, so that the bacteria will grow to make sure there is no bacteria at all.
I see. You're testing it.
Testing it if there is still any contaminants. And when we get a pure culture without any bacteria, we streak it on a agar's land like this. So this is a pure culture. And once we streak it, this can be here for 6 to 8 months without further subculturing.
And universities can do that too. But what universities haven't done is they haven't got any ponds. See,
they're still at the test tube stage.
Right. We have spent maybe $2 million getting to the test tube stage, and so have a lot of other people.
Yes.
But we have ponds of it growing outside.
Yes.
And so we're 10 years ahead of anyone else. Now, it may seem a lot of money to get to a little pure culture.
Yes, it does.
And Sebastian only gets his salary. But there's a lot of other things that go to make that $2 million, like the liners in the ponds and the piping, and each pipe costs $150, and you have to make 1,000 feet of it, and there's hot air, and there's harvesting machinery, and there's boilers, and
well, now there's got to be a major step between the test tube and the pond. If all these other people are experimenting,
that's the big step.
Okay, what do we do next?
Well, after you get it in the pond, then there's another step that we're 10 years ahead of most people, and that is that how do you get it out of the pond and get it into a powder that's a standardized product that you can buy in the shop?
That's clean and pure, and is the spirulina. And
yeah, because you know, your white flour that you eat has 100 parts of insects in it. The moths and things that go in the field, and little eyes and feet and stuff.
Oh, wonderful.
That's 100 parts, but our spirulina is pharmaceutically grown. It's pure. And,
I see.
Although we may have a few moths that fly into the water, and so we extract all that. And ours is so much cleaner than white flour. I mean, it's incredible.
Well, can you use spirulina flour like you could use white flour?
Sure you could. You can make it into bread, but it's so concentrated.
It's a plant, just wheat's a plant. In other words, the same idea?
You don't need 2% of spirulina in a product to make up enough nutrients. You know, you wouldn't want to eat more than that.
It'd be too rich.
Too rich.
I see.
Very concentrated. This is space food.
Where do you go from here, Sebastian? What's the next thing we do?
From here we go to a small plastic like this.
Well, we started there, and then we went to that, and then to here, and then back to a flask?
Medium. All this medium is sterilized, so there won't be any bacteria in the medium also. This flask is sterilized along with the medium.
Is the medium distilled water?
It can be tap water. You can use distilled water, but it again becomes more expensive. But spirulina can go on in ordinary tap water. You take the tap water and put all the nutrients.
But don't you have bacteria in tap water?
No, but you are going to sterilize it.
Okay, okay, that's what I was asking.
Yes. You sterilize the whole medium. And
then you get into
this. From here we introduce to this flask stage. It takes 5 to 6 days. Then it comes to a stage like this, to the next flask.
And then it gets
further, it goes to a 500 ml.
Well, why are these all different colors? Do you put dye in the water?
So all different types of spirulina. It depends on their phycocyanin content. And
phycocyanin is a
blue
pigment.
From the test tube to this stage, it takes almost 3 weeks. And from here, we go to a flat flush stage, which is on the other side of the lab where we'll take you.
Let's move over there now and take a look at that.
See, there are 10,000 different kinds of algae and diatoms, and Spirulina is only one of them. Would you like to point out the spirulina there? That's one of 10,000 kinds, and we have had to look through thousands of kinds to find out what are edible and what are nutritious. And of course, we found that spirulina was more nutritious than any other plant or vegetable on the planet.
These are photomicrographs of some of the spirulina we isolated in this lab. This one we isolated from Lake Ngorongoro in Tanzania. This one is from Ethiopia. And this one we got from a lake in Jamaica. And this one is from Lake Bogoria in Kenya. And this one from Lake Chad.
So every day they take slide cultures of these algae from the ponds, from out in the field. And they put it on a slide, and then they put it in this microscope, which is a very powerful one, a very expensive one. And you can tell from this exactly what's happening, and you take a look and you can see what other algae are growing, if any, or whether the pure culture has been adulterated by anything. And you can see by how the algae is growing, whether it's expanded or whether it's contracted, whether it's under stress from too much light or too much pH or some other factor. And these are different kinds of algae from different ponds. This is spirulina, this is Dunaliella, which is 100 times smaller than spirulina. And it has a little flagella with like oars which make it swim. And so it navigates through the water to get its food, whereas spirulina is a plankton type, which means it drifts in the water to get its food. So one is aggressively seeking nutrients, and the other just takes the nutrients that are in the environment around it. So one is called a planktonic type, and the other is something that it goes and gets its food, seeks its food. And these are just some of the instruments we use for looking every day at this. And looking at the optical density, we have other instruments for measuring how much algae is in the water, how much light is penetrating through the algae, and so forth. You want to say anything here?
No, are we going to the other section?
When you look through that microscope, how many times is the magnification?
Right now it is around 600 times magnified. So we can go up to 900 in this microscope. And another thing is, this has got a chamber, and you can grow single cells in this. There is an incubator, you can adjust the temperature and light, and there is an instrument by which you can manipulate and take one single cell out of this and grow it, and you can study the growth rate right in this food.
And we can take pictures of anything we're
Yeah, with these
looking at.
cameras, the photomicrographs.
I see that you have at least 7 different colored, would you call them flasks here?
Yeah, they're culture flasks.
They're culture flasks.
We take the pure culture, as you saw earlier in the test tube, and we grow it out. And this is the first stage before it goes out into the field, into the inoculation ponds.
So we've come from the smaller ones that we saw before, and they gradually come here, the culture.
It's from a test tube, and we keep adding medium into this. And this is spirulina. You see at a very early stage. Now, in 2 or 3 days, when you look at that, it'll be a very deep green. You won't be able to see through it at all. This is Dunaliella, which we have out in the ponds, and that is also an oil-bearing algae. It has about 29% oil in it.
Is this the one that you said was high in beta-carotene?
That's true. This is the highest source of beta-carotene in the world. It has about 6%. Spirulina only has about, maybe 1% or 0.9, or maybe going up maximum of 1.5%.
Now, I noticed there are dates on these flasks. 2 dates, what, about 8-10 days apart. What does that mean?
That's the starting date of the culture when we put it into the medium and start it off, and then as it increases in density, we keep adding medium to get more. And this is Nitzschia, which is another oil-bearing algae. These have all been studied by the government. They spend a fortune at universities encouraging the growth of this to try and stop importing oil. Because if we could grow thousands of acres of this, then we could extract the oil from them. And certain of these, like for instance Enchistodomus, that is up to 32% oil, and it grows in fresh water. The one next to it, Dunaliella, grows in 30% of salt water, which is equal equal to the salt of the Dead Sea. Or
So you have a different medium in each of these flasks.
Oh, yeah.
Does the medium explain the color?
No, it's
No.
the color of the algae for this
Yeah.
one.
In different parts of the world, you have lakes which are full of minerals. And like the mineral lakes of Africa, which are full of sodium bicarbonate, grow spirulina. And Dunaliella happens to grow in very dense salt waters. This particular species. And Dunaliella will grow in seawater too, and so will spirulina. We can grow it in seawater.
Now, the two green ones, the blue-green ones, are the spirulina.
These are, yes. They're a very light, early stage of the culture, and it will get a very bright green, and this will go into brighter yellows and brighter reds as the beta-carotene develops.
I see. Now, the next step from here,
That's the first pond outside, the first inoculation pond outside. We have hundreds of flasks like this, and when we have a certain ratio, we transfer it to the first inoculation pond, which is around 6 square meter in surface area. So we require almost 1,000 liters of culture like this to transfer to the first inoculation pond outside. Those cultures we grow in all these flasks.
then from the inoculation pond, you go to the regular
step.
we pond?
We go to a 25-meter pond, and then we go to a 50-meter pond, and then we expand it to a 200-meter pond, and then we go to a 600-meter pond, and then we go to 40,000 square meter pond. So,
Oh, well, let's go take a look at the ponds.
yeah, they're all out there. We're the only people with actual ponds of Dunaliella that's grown in universities and test tubes and tiny little raceways, but we have proper ponds with it in.
Excellent. We're standing in one of the spirulina ponds, and as you can see, it's very large. There's a plastic liner, and then the water is in here, and the spirulina grows. Do you always have to dry it out to harvest the spirulina like this, or do you usually not let it dry out?
No, we just let the water dry out of this pond last week because we're repairing the liner at the other end of the pond, so we wanted to drain the pond. So that the spirulina has dried now in a wafer kind of form, just as it dried out in the deserts when the Israelis found it in ancient times and called it the manna from heaven.
This is the original manna.
The same stuff. If you take a picture of this, you'll show them what it really looked like. If you go to the Bible in Exodus, you'll find a perfect description of how it looked white, just like this, and how it was highly nutritious. And you can even see a tree of life design in it if you look closely. Beam in on these antlers here. See a tree here?
Now, if we pick this up and ground it up, it turns like into flour. You can make bread out of it.
Sure. Or you can eat it just like a biscuit or a wafer. Or, I'll take a piece. You want me to lift a piece?
Yes.
Bread.
When you know you could.
I can take a really big piece.
There's a big one there that's separated a bit. We have, it's white on the top, and you can
It's a bit sandy on the bottom because we had a sandstorm here and blew sand in, so naturally it's got sand on one side. Normally you wouldn't have that.
So it would be almost the same on the bottom as it is on the top.
And you can separate the sand by brushing it off. In Africa, when they dry it, they actually put a puddle of it on the sand. So it dries with sand sticking onto one side of it, and then they just scrape the sand off. That's how they harvest it.
And then you could just eat it.
Yeah, you just eat it.
This is the original manna from heaven.
Yeah, it's the most nutritious stuff on the planet. And the Israelis lived on it for 40 years in the desert.
It was their source of protein.
It's a strange, there's a perfect description of it in the Bible. And of course it doesn't rain very often in the desert, but when this catches the spring rains, it all springs back to life and becomes green again.
So it could grow almost overnight.
Yeah. And
except on the seventh day, of course.
Well, I don't know. This stuff is interesting because if the dew comes
There isn't any dew here because the desert is very dry, but if we do get a moist dew night, the next day the flies will come and they love it so much they lay all their eggs in it, and that's just the description of what is in the Bible. But if they don't pick it before the dew, it's destroyed. See, it's high protein. There's 70% protein in this. So naturally, the flies will like it. So
Any living thing seems to know.
Any living thing, it will eat it. The dogs, we got problems with coyotes you wouldn't even believe. We try and grow it in tubes, they come and eat a hole in the tube to get at it. And we're now having to look at special smells that chase coyotes away because they eat a hole in the liners.
I see.
They love it so much. It's incredible stuff.
One of the things that you mentioned to me that impressed me today is that you said you had some people who were so thrilled with this particular product that they've been working as volunteers with you here at the research center. These are people with degrees who probably could go out and command a fairly sizable salary in industry, but they're working here because they've caught the vision of feeding the world.
Yeah, we have people even coming as far as from New Zealand. They have degrees in geology for earthquakes and all that sort of thing. And we have people with social science degrees and degrees in literature. They come and they work in the fields and things. Here, the employees start at about 6 o'clock in the morning to work, and they go home early in the afternoon. But the volunteers, they just get their board and their keep and enough salary to keep going, and they work till dark.
I'd like to meet some of these people, if I might.
Well, they're great, wonderful people. They've been with me sometimes up to 10 years because they know that they can get fulfillment from this. And I think we have some people right here in this place if you're interested.
I would like to meet them, just say hello to them. One of the things that intrigues me is I've been writing a series of articles on disasters. You mentioned one man who had written about or had studied earthquakes. And I know the disruption that could go on in our society if there were such an event. People could use spirulina to feed the crowds until the services got restored.
Yeah, I believe so. I think it would be a wonderful way of storing the sun's energy on the shelf until you needed it, and to be able to live off it for a long time if you need it. I'm not supposed to say it's a complete food or that you can live on it, but I believe myself that that's the survivalist answer. It's because it's a lot of concentrated food in a small amount of space, and it lasts forever. There's no
the shelf life is indefinite.
Shelf life is indefinite.
Excellent. May we meet some of your friends?
Sure, yeah. This is Phil Allen. He's been with me for about 8 years or so.
Phil, what got you interested in spirulina?
Well, that goes back a long way, but basically feeding the world and bringing peace to the planet is the most important thing to me.
And you think there'll be peace if people are fed?
That's the first step.
That's a good step.
And this is Richard Welker. He's got a degree from San Jose State, and he sort of runs things around here in the field. He's a field manager.
And what got you interested?
Well, I've known Dr. Hills for about 10 years, and what really got me interested in spirulina was when Dr. Hills bought 300 tons and said, pack it in the warehouse. And
300 tons?
That's right. And we had a most amazing experience. I think an article occurred in the National Enquirer 8 or 10 months after he bought the spirulina, and the distributors were suddenly banging down the doors when they found out that they could sell spirulina for weight loss. And we had a terrific time trying to keep the inventory up and so forth. So that's how I really did get involved with spirulina, and it's been an ongoing love affair since, I think.
I see. Dr. Hills, thank you very much for being our guest today. I think all of us have learned a lot about nutrition and about the new plant that's the old plant that's been restored.
Yes, I do believe that now is the time when it's going to heal the planet and be an instrument for the light, because it is a way of harnessing the light's of the sun, the light of the sun, which is an inexhaustible source, and a way of putting it into your body, too.
Yes. Thank you.